Human blood contains many different components, including numerous different types of white blood cells. Some of these include lymphocytes, monocytes, eosinophils, basophils, neutrophils, and others. Lymphocytes are further subdivided into T cells, B cells, and NK cells. Distinguishing and separating different types of these cells is highly important in carrying out research in the field of immunology. Currently, this is done by staining the cells using fluorescent antibodies that target specific receptors present on each type of cell. For example, the T cells and B cells express CD3 and CD19, respectively. The stained cells are then analyzed by flow cytometry according to their size, granularity, and fluorescence. It has been long thought that it was impossible to distinguish different immune cell types without using these antibodies. However, a new discovery made within the Institute for Basic Science may have just changed this view. Our researchers discovered a new fluorescent dye that only stains B cells over T cells, without targeting any cell receptors. This new dye is called CDgB. It is a lipophilic molecule that contains a polar fluorescent component attached to a nonpolar hydrocarbon chain. This means that the free unbound CDgB dye molecules form aggregates similar to micelles in the solution, which suppresses their fluorescence. When they attach to the cell surfaces, however, the aggregates dissociate and become highly fluorescent. The lipophilic nature of the dye means that the dye does not bind to a protein target, and instead localizes to the lipid membrane structure directly. The researchers optimized the dye by testing different hydrocarbon chain lengths, from 4 to 20 carbons. It was found that the CDgB with 18 carbons showed the highest selectivity towards the B cells. Our researchers investigated why this dye favors B cells over T cells by performing lipidomic analysis of the cell membranes. It was found that B cells' membranes had shorter phospholipids than that of the T cells. In addition, the membrane cholesterol content in the T cells was about 2 times higher. These factors give the B cells a more flexible cell membrane, which is why the CDgB molecules attach more readily to these membranes. Even among the B cells, it was found that the strength of the fluorescence was different based on the cell maturity. The B cell progenitors and immature B cells gave off much brighter fluorescent signals than mature B cells, which is most likely due to the higher membrane flexibility in the immature cells. Our researchers called this selective staining of different lipid membranes a lipid-oriented live cell distinction technology. They also stated that CDgB was the first example to report such type of cell distinction mechanism. This video was provided by the Institute for Basic Science. Please support us by liking our videos and subscribing to our channel. For more information, please visit our homepage, Facebook, or Twitter in the links below.